Hello and welcome to the online version of Integrated Pest Management. My name is Todd Gaines. I'm a weed scientist in the Ag Bio department, and I'm going to be speaking today about principles of pesticide resistance management, particularly as they relate to IPM. First of all, we need to define pesticide resistance. The key thing is that this is a heritable characteristic of a pest that makes it less sensitive to a pesticide. So that's implies that this is an evolutionary process. It involves a genetic trait in the pest, and this trait allows the pest to survive the pesticide in a way that that treatment would normally kill individuals of that species that don't have the resistance gene. This occurs in all types of pests, including weeds, insects, mites, fungi, bacteria, and ultimately, why does this matter? It means that the product that you're intending to use to control a pest is not going to work. You're going to have a control failure. This is based on a mutation generally in the target site gene or in a gene that can somehow provide a protection to the pest against the pesticide. And whatever this mutation is, it's genetic and it's making that pest less sensitive to the pesticide. These can naturally occur in the populations before the pesticide is ever used. So you know the pesticide is going to kill susceptible individuals, those that have the sensitive or allele and that don't have the mutation, and it will select those that have the mutation to survive. So those that have the mutation reproduce, pass on the genes, and uh, then that gene, that allele, increases in frequency in subsequent generations. There's another term, tolerance, and even though these two words, resistance and tolerance, kind of can have a similar implied meaning, tolerance does have a different definition biologically, and that it's talking about the natural ability of a species or a member of a species to survive and reproduce after a pesticide treatment. So this implies that no selection has happened to make the organism this way. It's a natural tendency. So for example, this can be developmental. Mature caterpillars are tolerant to insecticides that would control the same species when it's younger. So this is due to differences in size, thickness of the exoskeleton, metabolic rate. So that's identified as tolerance rather than when we talk about resistance, it implies there's selection. Individuals with different genotypes are surviving differently. Another example of tolerance you can see for weeds at the bottom is that some species can simply survive and reproduce after herbicide treatment. So that means there's been no selection or manipulation to make it tolerant. It's naturally occurring. An example of this is that corn is naturally tolerant to the herbicide atrazine. No selection of any kind has happened. All corn is tolerant to atrazine through metabolism. But then a number of weed species might have been selected for resistance to atrazine over time due to genetic variation. And that's the definition on the top. This is the Weed Science Society of America definition of resistance, showing that it's an inherited ability of a plant to survive and reproduce following exposure to a dose of herbicide that would normally be lethal to the wild type. When we're talking about entomology, this is a definition I really like. So this definition is a genetically based decrease in susceptibility to a pesticide. And going further to talk about something called practical resistance. So this is field evolved. That means it's happened out in the real world. And it is a resistance that reduces the efficacy of a pesticide and has practical consequences for pest control. So sometimes the debate will get kind of caught up in, well, okay, so this population or these individuals are resistant, but is it above the field rate? Does it matter? And that's where you kind of come into this definition of practical resistance. It means, yeah, it matters to whoever's managing it. They'll notice it. It will be a problem. Now here you can see, this is just a diagram kind of showing differences between lab selected resistance and field selected resistance. And then within field selected resistance, there might be some cases that just aren't a big deal because the application rate is higher and it can be managed. But practical resistance means, that product as it's normally used doesn't work anymore. Finally, you can see the definition of resistance for a fungicide, and that's, you can see here, a stable heritable trait that reduces sensitivity to a fungicide by an individual fungus, emphasizing this as an evolutionary process. In particular for fungicides, there are some that have a multi-site mode of action as opposed to a single-site mode of action. So those with single-site modes of action are at a higher risk for resistance compared to those with multi-site. And typically, most fungicides and all pesticides in development today tend to have a single site mode of action because this reduces the chance for unintended off-target effects. So how does resistance happen? There are some certain characteristics of pest species that make them more prone to resistance. So this can be, they make a lot of progeny, they have a high fertility rate, they might reproduce quite frequently. Think of an insect species that might have 5 or 6 generations in a growing season, compared to a weed that might have 1 generation per year. So there are a lot of biological characteristics, but the selection pressure is part of the evolutionary process, and that's what's under management control. So how much of the pesticide are you using? Certainly, resistance is a dynamic system. So you have an interaction of the pesticide itself, how it works, how the pests are exposed to it, these characteristics of the species itself, so its life history, how it reproduces, how many progeny, its genetic diversity. But then this management part is where, again, of course, as the IPM expert, you have the ability to intervene. So you can look at economics, you can look at risk, you can look at all the factors that go into the decision making of which pesticides are used and when, at what rates, and how much. Ultimately, you can never really prevent pesticide resistance if you're using, the only way is to not use the pesticide, but you can do things through management to delay when resistance would become a problem and try to delay that for as long as you can. Specifically, some genetic factors affecting resistance evolution include frequency of mutations for resistance, whether or not resistance is dominant. A dominant resistant trait is going to increase in frequency more quickly than a recessive trait. The fitness of organisms with the resistance allele. So if there's a trade-off that the resistance mutation causes them to grow less quickly, for example, that can slow down resistance. And the number of genes involved. A single gene trait will increase more quickly than a multi-gene trait. For the species itself, you have ecological and behavioral factors such as reproduction rate, migration, how much gene flow is happening. Do individuals move between sites each year and between generations or not? Are they sessile? And how are they actually exposed? Do they have to feed on the pesticide? Is it something that the spore needs to land on the leaf and get exposed to it, or is it systemic in a plant? These are all important factors. And then operational factors can include whether a pesticide has a relationship to earlier used pesticides, such as do they have the same mode of action? Does it have a residual activity that might degrade over time? And how is it applied? How directly uh, applied to the organism is the method. This is a diagram that can kind of just give you graphically a way to show how resistance can evolve. So imagine this is an initial population and the individuals in dark red are homozygous for a resistance mutation and the kind of pink or light red ones are heterozygous for a resistance mutation and the ones in white are susceptible. And you'd assume that initially the resistance trait is going to be quite rare. So then you have the pesticide application and every individual that's susceptible dies. And in this case, the heterozygous ones die, only the homozygous resistant ones survive. But now, unfortunately, those are the ones that are going to reproduce and they have the genes that make them less sensitive to a pesticide. And now progeny are going to be only homozygous resistant, right? No susceptible ones had a chance to survive and reproduce. The humans, due to this selection pressure, have made this happen. And now this new population is more resistant than the natural population. Now, in a real scenario, there might be new susceptible individuals that would come in. There might be a seed bank or a reservoir of susceptible individuals. And, a number of factors make it so it doesn't necessarily go from susceptible to resistant in one generation. But this is what you try to understand when you study how this will occur. And eventually, the population is going to be mostly resistant individuals. That resistance mutation will be very frequent. And when the selection pressure keeps going, the resistant individuals outnumber susceptible ones, and the pesticide will no longer be effective. This is an example of the proof that these resistance mutations really do exist in populations before the herbicide's ever used. This is a weed called blackgrass. It's very common in Europe in wheat. And the scientists went to old herbarium collections that were taken between 1788 and 1975 before any of these ACCase herbicides that have ever been used, and they actually found a mutation that confers resistance to these herbicides in a sample collected in 1888. So it shows that the resistance trait is out there before herbicides are used. The pesticides don't cause mutations themselves. It's part of the standard genetic variation in pests. This is what resistance can look like in the field. Here you can see this is a weed called horseweed that's been sprayed with the herbicide glyphosate. There are a lot of susceptible individuals that have died, but here's some resistant ones that, or susceptible ones died, the resistant one lives. So you imagine that this one is going to make seeds and progeny, this one isn't. So the next generation will have a higher frequency of resistance. So why is this important? Well, certainly pesticides are a very important tool when it comes to our overall program and options for managing pests. And pesticide resistance is increasing. You can see the hundreds of species of arthropods, weeds, plant diseases, rodents that have resistance to some type of pesticide registered to control them. And there just aren't many new pesticides being discovered and commercialized. Rightly so, we have very high regulations for safety for both to the applicator, to the consumer, to the environment. And so it can be challenging to find a pesticide that's both effective only on the target species, selective in the crops, and safe and doesn't have a long life in the environment. So it means that the pesticides that we have, it's very important to preserve their activity from a pest management perspective. Here you can see the increase in rate of evolution of resistance to a number of different herbicide groups over time. If you're interested in more, this website weedscience.org is a great reference to find out all the current information you'd like to know about herbicide resistance. Again, pesticides are important for effective pest management, and it is difficult and a lot of barriers to develop new products. New things do come out, but that's not the answer. So we don't want to run out of the products that currently work. And as well, we don't want resistance to happen in one place to evolve and then spread to other locations, right? Because the organisms themselves can be mobile and spread resistance other places. And ultimately, if there's resistance and the pesticide's still being used, it's not an effective use. So there's money that's being wasted. This product is going out in the environment unnecessarily. So there's just a lot of reasons why delaying resistance is important. This is one of the most major weeds in the US and increasingly in South America. It's called Palmer amaranth. And you can see here is evolved resistance to glyphosate or Roundup, and it can really take over a field, produce an incredible number of seeds. It's basically one of the fastest evolving species for herbicide resistance. Again, here are some websites for information, resources on pesticide resistance. You can see the HRAC, FRAC, and IRAC. These are agrochemical industry sponsored groups with their great information on the classification of pesticides and uh, as well as resistance management information. Just to further show that resistance is an evolutionary process and it's all about the selection pressure and a lack of diversity in management, this is an example of resistance evolving to hand weeding in a weed called barnyard grass. So if you look at this photo, on the left is rice, and on the right is barnyard grass that is normal. So you can see it has a red stem and droopy leaves, whereas rice has a green stem and upright leaves. In the middle, this is barnyard grass, but see how it looks just like the rice. It has a green stem and upright leaves. So this is a genetic trait that's been selected over time to escape hand weeding. Now, so why are some products more resistant prone than others? All pesticides have some risk, but some are classified as higher risk than others. And this generally comes back to the single site mode of action and more targeted. So here's also a look at sort of low risk versus high risk uses of pesticides. Everything on the low risk end has in common that less pesticide is used. That's a very general principle. The less pesticide you use and less frequently, the less selection pressure you're going to have for resistance. Whereas the more you use and the more frequently, the more you'll be selecting for resistance. So you can compare a lot of these on the spectrum. It's very informative. So on the low end, alternate applications of different pesticides. On the high end, repeat applications of the same pesticide. On the low end, base your spraying on an economic threshold. On the high end, just spray on the calendar, for example, every 2 weeks, regardless of what's going on. On the low risk end, have a crop rotation so that you're breaking up the life cycle and the timing of what's growing in the field. On the high end, a continuous crop that's predictable and the same every year. On the low end, spot perimeter treatments only where it's needed, as opposed to treating the whole field at higher risk. So let's come back and talk again about the mode of action. So we're talking about comparing pesticides for higher and lower risk. What can you do as management? So the chemical structure of the pesticide directs what its target site is and how it works. So the target site is the physical location within the organism, the actual enzyme that's inhibited. And the mode of action is everything involved in how that pesticide gets there and how it causes some kind of physiological disruption that leads to death of the organism. So pesticides are classified by this mode of action, and each one has a group number that helps managers make resistance management decisions. And this group number is marked on the labels, and you can also go to those websites that I mentioned, HRAC, FRAC, and IRAC, to look up all the available insecticides and what their mode of action is. Within herbicides, there are around 22 modes of action, insecticides 27, and fungicides around 40. And these are all affecting, various aspects of the organism development, growth, and particularly synthesis of things like amino acids and lipids, as well as respiration. Now, again, why does this matter? Well, what you can do is rotate between these different mode of action groups, as well as have mixtures. But it's essential that you know what the mode of action is of any pesticide that you're using, so that you can be sure that subsequent applications don't use the same mode of action, or again, you can use mixtures. So this is an example of an insecticide called Lorsban or chlorpyrifos. Now this one is kind of famous recently for other reasons. It's been the subject of a lot of regulatory scrutiny around its safety. But just from the mode of action perspective, you can see that it's called a group 1B insecticide, which is an organophosphate. And you can see that all the registered insecticides listed in this group, and that they're all acetylcholinesterase inhibitors. You can go to the IRAC website and learn what is an acetylcholinesterase inhibitor. And so you can see here that this is inhibiting acetylcholinesterase at the nerve synapse and sort of overloads the nervous system. For fungicides, so they have their own numbering and lettering system, but again, it's based on cross-resistance behavior. So you can see here this Ridamil Gold is a group 4 M5. So they have an FRAC code, and then they also have this kind of mode of action system as well. So it's a bit complicated, but you can look up this group 4, and so you can see that these are inhibitors of RNA polymerase 1, and that resistance and cross-resistance is well known in various mycetes, and that it's considered high risk. So look at these guidelines for resistance management. So again, a really nice resource to look into for more information. For herbicides, it's a numbering system that's used by the WSSA, and the HRAC has just in the last month come out with a new numbering system. But again, they're based on site of action. So you can see here, group 1 is called, this is for these ACCase inhibitors. So it's a form of lipid synthesis, and you can see all the herbicides that are registered with that group. There's some more important terminology for you to know. Cross-resistance is something you might hear a lot. So this is when resistance to 2 or more pesticides results from a single resistance mechanism. And it's not necessary that the pest would have been exposed to all those products. So this is why cross-resistance is really a problem. So an insect maybe has been selected with parathion, which is an organophosphate. Then it would also be therefore less susceptible to the other organophosphates that are also within that same group. So it's not always the case. It might, some resistance mechanisms might only give resistance to one specific pesticide molecule, but others could have this cross-resistance. It could also cause cross-resistance to a different mode of action group. This is different from multiple resistance. So this is when resistance to several pesticides results from two or more distinct resistance mechanisms in the same organism. So for example, Palmer amaranth can be resistant to atrazine and also to mesotrione. These are two different groups and two different mechanisms. From a management perspective, or just looking at it, it's kind of hard to know because you see that it's resistant to both groups and you don't know necessarily if it's the same mechanism or not. But the cross resistance is kind of more of a problem because you might get something unpredictable. You might think, oh, okay, I've used a group 1 and now I'm switching to a group 4 and I shouldn't have resistance to that. But if there's cross resistance that you don't know about, you would. So that's just a layer of detail that you may need to deal with at some point. Certainly multiple resistance in species is increasing. So you can see here the increase in weeds over time, resistant to 2, 3, 4, 5, 6 different modes of action within uh, one population. So that's really a problem, when nearly every registered product you have resistance to. In Illinois, water hemp is a major issue, and you can have populations actually that are resistant to, say 4 different modes of action or even 5 or 6. And again, it just highlights the need to sustain the available resources for management. Other terms you may come across include qualitative resistance. So this is basically qualitative is single gene and it's yes or no, either it works or it doesn't. So basically, if you have qualitative resistance, it just doesn't work. There's no control. Quantitative resistance, there's going to be kind of a range in sensitivity, more gradual, and you might be able to increase a rate or do a mixture to combat it and still control individuals that have quantitative resistance. Overall, pesticide resistance management is this process of limiting or delaying the evolution of resistance in a pest population. If you're using the pesticide, there's no way to prevent resistance, but you can slow it down, delay it, limit how much of a problem it would be by doing various things to essentially use less pesticide. Again, that's the goal, is delaying evolution of resistance, and then that's a proactive approach. But you may have cases where you've detected a resistant biotype, and you can do things to try to reduce its frequency or reduce its impact as well. They're all part of IPM, and one way is to minimize at-risk products or use them as infrequently as possible. Certainly, to the extent that you cannot rely on pesticides alone, integrate different controls, particularly integrating, for example, beneficial insects, predators, and parasites. You really need to think about the insecticides you're using so that they don't kill your beneficial insects, right? So a lot to go on there. You could have biological pesticides, cultural practices to make it an unfavorable environment for your target pest. There might be transgenic plants that either are expressing a protein that is a pesticide itself to the target pest, or allows you to use a certain type of herbicide, or the transgenic plant that might have a disease resistance trait to help you manage the disease. Crop rotations, different varieties of crops, so pest or disease resistant varieties, certain types of attractants, deterrents. With weeds, you can think about harvest weed seed control. So you have a whole, certainly IPM toolbox, and you want to think about how you can reduce the density and severity of your pest in as much as possible without using pesticides or, with minimal use, and that's going to reduce and delay your selection pressure. Now, when you think about these pesticide modes of action groups, anything you can do to mix them, rotate them, apply them in sequences among different pesticide mode of action groups was really helpful, and avoid using the same mode of action group over and over. And you've also got to be careful to really understand what the mode of action group is. Don't rely on product names and not just on active ingredients, because often a different retailer might have a different product name, but it can be the exact same thing. And here's an illustration. So these are fungicides for control of alternaria leaf spot and cantaloupe. So you have 5 different products, and okay, these seem like different things, but they all contain FRAC group 11 fungicide. And so, in this case, for example, going between Cabrillo and Quadris, is really not an effective rotation for resistance management because pyraclostrobin and azoxystrobin are both in group 11. Here's one that's a premix of a group 7 plus group 11. So that's mixtures are always helpful because if the pest is resistant to one, it shouldn't be, and it's not resistant to the other, you'll control it initially. So that's a good preventative resistance management strategy. And then here's one that's a group 3 plus a group 11. But again, you can see why it's worth it to look into the mode of action group and understand what's going on there. Typically, the label is going to have some informative resistance management guidelines. And keep in mind that because they're on the label, that makes them a legal requirement. They must be followed. And so, for example, when you're looking at these group 11s, they've have this requirement: do not apply more than one application of Quadris or any other group 11 fungicide before alternation with a fungicide that is not group 11. Also, do not make more than 4 applications of group 11 fungicides per crop per acre per year. So that's telling you, when you get to that level of selection intensity, you're going to be selecting for resistance. So therefore, make sure you're alternating to different groups. Certainly, everything you can do to make sure the pesticide is applied correctly is going to be very helpful for managing resistance. So that way, you're sure to target the most vulnerable life stage of the pest, use spray rates and application intervals that are recommended and in compliance. And particularly, the full recommended rate is generally helpful. That's you're still going to be selecting for qualitative resistance in that case, but uh, when you start going with lower rates, that's where you can select for the quantitative resistance. It's more kind of gradual, creeping, multigenic type of resistance. So high rate alone is not a resistance management strategy, but it is part of the right rate, the right time, and rotations and mixtures on as far as thinking about pesticides. There are certainly challenges. There may not be other things that are options for management. There might be not be other registered products to have a rotation or a mixture. So that can be a really problem. Your other options might not be as effective. Very common, the other options might be more expensive, and that can be a really big deal. And then you're not just managing one pest or one species, right? So it can often be the case that maybe you have resistance in one species, but you're also using that product to manage other species in the same field. So what do? If you're still using it, you're applying selection pressure for resistance in your one species, but you're managing other ones. It's a trade-off. Another core principle is anything that you can do to preserve susceptibility genes within the target population is very helpful. So you can provide a refuge or a haven. This particularly applies for insects. And so you might have unsprayed areas within a treated field. You might have adjacent refuge. The idea here is that the susceptible individuals will interbreed with any resistant individuals that survive. So it's going to dilute the increase in frequency of resistance in the next generation. And especially if only homozygous resistant individuals survive, any resistant individual that mates with a susceptible individual will have heterozygous progeny. And so you'll be able to control this with a high rate. GMO crops also fit into this conversation in terms of, the main types of GMO or transgenic crops out there are either for herbicide resistance, particularly Roundup or glyphosate resistance, or BT traits. So these are the Bacillus thuringiensis crystal proteins that are toxic to certain types of insects, such as beetles and moths. So for both of these, they're basically the the crops for herbicide resistance give you new uses for herbicides, but it's still the management practices and how you use the herbicide that are selecting for resistance. And for the BT, again, it's a pesticide itself, so you need to think about how you're using it. You can have mixtures, so now it's quite common that BT traits are stacked. So you have more than one BT protein expressed in a given crop plant. You can have refuges in how you plant the crop, even refuges within the field. So again, it comes back to the management decisions and in terms of how much of an issue resistance will be. So thanks very much for your attention. I'd like to acknowledge particularly Meg McGrath from Cornell University and the others involved. Some of these slides were taken from the Northeastern Sustainable Ag Research and Education Program to put together slides on resistance management. And feel free to reach out to me if you have any questions on this material. Thanks.